Case 6, mac and cheese overload. Philip's friend told him that it will take a long time for the liver to break down the high concentration of yellow 5 and 6 dyes from his mac and cheese, but Philip insists that his liver will just work faster to break it down. This lab tested the accuracy of Philip's claims. Our hypothesis is if the substrate concentration increases, then the rate of reaction will increase until a point of saturation. This is because as the substrate concentration increases, more substrate is available to bind with the enzyme and more product will be produced. However, after a certain concentration, increasing the substrate concentration will have no effect on the rate of reaction since enzymes will be saturated with substrate. The procedure was 1. A plastic tub was filled 2/3 of the way with water. 2. A graduated cylinder was inverted and submerged in the tub of water with all the air removed. 3. 10 milliliters of 3% hydrogen peroxide was measured out and was added to the bottom of the reaction chamber. 4. A paper disc was soaked in catalase and placed on the wall of the reaction chamber. 5. The chamber was stopped by the stopper apparatus. 6. The tube's opening was placed under the graduated cylinder. 7. The reaction chamber was flipped so that the hydrogen peroxide came in contact with the catalase-soaked disc. 8. The volume of oxygen produced, the dependent variable, was measured every 30 seconds for 10 minutes and data was recorded. 9. Steps 1 to 10 was repeated with 3 other test groups containing 2.25%, 1.5%, and 0.75% concentrations of hydrogen peroxide. Table 1.1 shows the makeup of the 10 milliliter solution used in each test group in terms of the total concentration of hydrogen peroxide. Each solution was made up of a certain amount of hydrogen peroxide and water. Table 2.1 shows all of the results of the data from this experiment. The initial data obtained from the experiment of each of the trials for the 4 different concentrations are shown, as well as their averages. The rate of reaction was determined by this formula: the final amount of oxygen produced in 10 minutes of each concentration divided by the time, 10 minutes. Here are our results from each test group. Figure 1.1 to 1.4, shown on the right, shows the graph data of the results obtained from the experiment. These graphs depict the trends of the relationship between the amount of oxygen produced and the amount of time. The rate of reaction was very fast at first and eventually slowed down in all 4 concentrations. In Figure 2, the rate of reaction is graphed, and based on the curve of best fit, the relation that results shows how the rate of reaction increases as concentration increases, but the rate begins to plateau around here. In this lab, the chemical reaction that occurred was the decomposition of hydrogen peroxide into water and oxygen. This reaction was catalyzed by the enzyme catalase. To decompose hydrogen peroxide, catalase catalyzes the reaction by lowering the activation energy of the chemical reaction by interacting with hydrogen peroxide through the enzyme cycle. The experimental results do agree with the hypothesis as shown on this graph. As substrate concentration increased, the rate of reaction increased. This was seen as more product was produced when the substrate was more concentrated. This is due to the increasing number of collisions between substrates and enzymes. When the concentration is higher, there are more substrate molecules to collide with enzymes, thus producing higher amounts of product. However, as the enzyme molecules approach the maximum rate at which they combine with the substrate, increasing the substrate concentration has a reduced effect. Eventually, the rate of reaction levels off. At this point, the enzyme molecules are saturated with substrate. Saturation occurs when each enzyme's active site is occupied by the hydrogen peroxide, and the rate of reaction becomes constant despite increasing substrate concentration. However, the reaction did not reach a point of saturation as stated in her hypothesis, but it can be seen that the trend is beginning to plateau as seen in Figure 2. As the concentration increased, the rate of reaction increased, but it started to slow down. There were areas for improvement that could be carried out to make the lab more precise. The controls in the lab could be controlled with more precision. For example, the amount of catalase on the plate could have been placed on the paper disc with a micro pipette rather than dunking the paper disc in catalase. Second, although the materials were not available, exploring higher concentrations of hydrogen peroxide could better display the exact point of saturation, as they would be saturated quicker. The significance of this experiment furthers the understanding of the natural world and products used in society, for example, hydrogen peroxide contact lens solution. Disinfecting the lens is based on the oxidizing effect of oxygen atoms released during the decomposition of hydrogen peroxide. It has been discovered that concentrations between 1 and 5% are the best for contact lens solution. Another application is preventing the browning of fruit by decreasing the amount of oxygen with which the fruit comes in contact. The enzyme catechol oxidase catalyzes the substrate catechol and oxygen, should produce benzoquinone and water. With the increase of the substrate concentration, the fruits such as apples will create brown pigments at a faster rate. This experiment raises other practical questions. For example, does the amount of enzymes also impact the rate of reaction? What is the concentration of hydrogen peroxide to reach the saturation point when the amount of enzyme used is kept constant? One follow-up experiment could increase the amount of enzymes with a constant amount of substrate to investigate the impact of different amount of enzymes on the rate of reaction.